Cool. Hi, everybody. It is November 3rd, 2021, our 7th week at Drexel fall term. So it is week 7 of the project. I wasn't here last week. I was in Chicago at the Neurocritical Care Society annual meeting with my co-op. And we basically went to show off our product at a trade show during that conference. And it was really cool to get perspective about the kind of technologies that's out there in neurocritical care. And I hope to talk more about my experience when there's, more of an audience. But for now, we can focus on things that are to do for the project. So I'm sorry, Bennett, we have been looking at predicting the reoccurrence of non-small cell lung cancer using this publicly available data set. It's called radiogenomics because it has imaging features as well as RNA sequencing data. So you can think of that as the genes that were expressed in the cancer. And then here's someone else. Oh, Alex is here. Cool. And then there's also the clinical data. Alex. Okay. He's loading. Okay. So yeah, we have this data set and we are working on the pre-processing of the data set, and particularly for radiogenomics, that is surprisingly a big part of it, because we need to extract imaging features from the CT images specifically, and that is not the code. Let me find it.
I mean, it's the biggest part of any machine learning project. Wouldn't you
Yeah,
agree?
but for some data sets, it's a lot simpler.
It's true.
When we think about the MINTS data set, it's pretty straightforward. Let me open up so you can find it. Likely I'll have to do a file manager. So yeah, we've been working on the pre-processing necessary to run really any model on it. And I think that Savik posted some interesting models that we can use. I haven't looked specifically into them, but we've been working on this script right here that utilizes two things that I kind of really like. It utilizes this open source lung segmentation model that was trained, I think, on segmented lung data. It's a UNet-based model that will take in a DICOM image of a CT scan of the lung, so like a cross section down the thorax, and return the segmented region. It's been pretty useful, so we don't have to rely on, getting those images manually segmented. It's pretty accurate. So I would, for anyone who's interested in actually segmenting lungs or CT images of lungs, I would rely on that. And then there's this package called Radiomics, which has all of these different categories of features you can extract given a pixel array, which is the lung, and then a segmentation. And Alex, your specific contribution to this was the process clinical features. So you have taken the spreadsheet, which is, let me see if I can find it. That's the.
I also documented what I wrote on the paper as well.
Okay, yeah, that's helpful. I think this metadata is for. This is the clinical features, I think. The metadata inside of this directory is describing what each image is, like what type and how many there are, because there's CT and PET, and then it's of different regions of the body. Yeah, so these case IDs are the individual people, and then you've encoded whether they smoked, how many packs each year they smoked. I think that's what it is, or something similar. So that's pretty helpful. And you did a ordinal encoder. We actually might want to think about doing a one hot encoding for these vectors instead of ordinal encoding for the categorical features. I like one hot encoding better than ordinal encoding, because I think the reason I like it is because when you're if you're looking at a neural network, if you think of how a neural network works, it turns off, it kind of forces the neural network to use a set number, a set number of neurons for one ordinal value, and then a set number for another. So if you have, like, if you're looking at one feature and the one hot encoded vector, like if we're talking about like dog, cat, and horse, right? This is dog. Only the first neuron, if you have like 3 neurons in the first layer, the first neuron will be activated here. But if you look at cat, right, it's going to be the second one, and then horse is going to be the third one. So it kind of forces certain neurons to be used depending on which categorical variable which category in the variable you're actually using. I don't know. There's always preferences to encode the data.
Yeah, because it's a little bit like slightly subjective for some of the ordinal features. Like, for example, survival status. I feel like it's subjective whether or not like someone being dead is ranked higher or lower than someone being alive, but I feel like there should be a difference somewhere. But the ordinal encoding kind of gives it, I think, randomly by default. So like it'll just set first one
Okay.
first, something like that.
You can try both, because
Yeah.
I think I usually reserve ordinal encoding for when there's a hierarchy of, like, if we're like giving ordinal encoding through 1 through 5 ranking, where 5 is the best and 1 is the worst, that makes sense to me. I wouldn't one hot encode that ordinal ranking, because 5 is obviously better. 5 is higher than 1. 5 is better than 1. Yeah, but that's good though. So this function will do just this function. I prefer functions to not have underscores, but we can fix that later. The last thing is really this function. So what this function does is it uses the root directory and the metadata in the root directory to calculate features for every image for every patient. But the thing is, and I've discussed this before, we don't necessarily want to do that, right? We want to find the CT image, the slice that has the largest area, because that's likely to, that has the greatest likelihood of having the most telling features about cancer, because it has the most, it has the most area, so most room for cancer to be there. And then we're going to select the 5. We're going to select 2 CT images before, 2 CT images after, and we're going to use all those as features. And I think the PyRadiomics package calculates 161 features, so we'll have 5 times that many features per patient. Yeah, that's the idea. I actually, in the Slack, I think I should have done this a lot sooner. From doing these projects, I've learned 2 things. One, find a data set as soon as possible. And then just recently I've learned to make a small reproducible a subset of the data set as soon as possible as well. And I didn't do that until last week. So what I did is I took one of the patient files and put it right here. It's only 200 megabytes. The whole data set is about 100 gigabytes. So that isn't great, but this encompasses everything you need: the genomics data, the clinical data, and one patient, right? And I just want to delegate that to somebody, because I could do it myself, but A, it's good for you guys to learn, and B, I'm pretty busy with work and other things. But if worse comes to worse, I can always bust it out. Does anybody have any questions about that? The code?
So you're saying we should try to run the code on our own directory, right?
Yeah, I would try to do that. Yeah, I would try. I think the main function uses this root directory, which is mine. So if you just put what I gave you in, so this points to NSCLC, and this is NSCLC small. So if you just route this to that small version, then it should work. I'm saying should. So I may have messed up, like copying and pasting the file names, but someone will have to yell at me if that happened. But yeah, the point of having this is so that it's reproducible, and B, it's a small set. And then I implemented one basic function, which is just the run to random forest. And I actually tested this on just the genomic features, so the RNA sequencing data. After I removed the non-variable features, I think it only, I don't know, it didn't do, the model was basically worse than a coin flip. So I wouldn't worry about that. But I think when we have that with other features, maybe, I don't know, it'll perform better.
So it got very low accuracy. Is that right?
Yeah, I think it wasn't necessarily that it was low accuracy. It was just, I think in the training set, I only had, our sample size is also small. Like, we're only dealing with these patients because these are the patients that have the genomic data. And what that means is that there's a very high likelihood, there's a higher likelihood that all of the patients in the training set will belong to one class. So I think what happened is that the training set only saw like the ones that weren't smoke, or the ones that didn't reoccur or something like that. And then it predicted that. And so the sensitivity was like 1%, the sensitivity or specificity was like 1, and because it only predicted one value. So obviously we want to be careful how we, create a composition for our testing and training set, so that doesn't happen. I don't know how that happened, but it was odd. But that's something we have to work on. So besides that, you can run it, and then in this function, we'll need to develop a way. You'll likely need to do is you'll likely need to have a function, a for loop like this before it, or maybe for each. We get a question.
No, that's just for
No, that's
testing something.
Yeah. We'll do a survey of models. Yeah, so the likely what people will need to do, whoever wants to take this on, is inside of the for loop that loops over all of the images for one patient, all the CT images for one patient. So I imagine that's like right here, I think, because there are multiple imaging. There's like two. There's the patient, and there's an imaging directory. There's another imaging directory, and then there's the CT. There's a DICOM images. So it's likely that you'll need to loop through those images, find a local max, or find the global max for that patient of the volume, which is in one of the features in the 3D features function. It's one of them. If you look up radiomics shape, radiomic shape, I can show you. It'll tell you which feature is the shape. So if I go to 3D here, mesh volume, voxel volume, surface area. This is the feature we want to maximize right here. And we're gonna just calculate that for all images for one patient, find the maximum, and then just calculate the features for the 5 images where the maximum is the midpoint. So you're gonna you're gonna find the index of the max before calculating all the features in that loop. Yeah, that's how I would do it. I don't want to store all the features and select them afterward. I'd rather just find the index for a patient, calculate the features for that patient, store the features, go to the next patient, which works well because we only have one patient to test, and then I have the rest of the patients. So the second part is the paper. Oh, good. You did add it. I can add, what I'll do is add, that'll be my job. Add segmentation. Let me put my name, Ethan. Add in, what's a comment in here? What am I missing? Oh, is it? It's difficult dealing with a lot of programming languages because they all have their own, like,
Yeah.
I've been working with like HTML, and that's different than JavaScript, and that's different from Python, and that's different from LaTeX. I'll put my name down for this because I did that. And then
do you use LaTeX at all at your job? Because you
my
said
job?
you said you were software engineering, right?
No, well, for the research I do, it's funny. So when I submit to conferences, they actually prefer Word because it's more, it's easier to configure. Although it's not entirely reproducible because often they offer templates in Word. For my job, I do my abstracts through Google Drive, and then I take it and submit it into Word. It is a lot of work to create a LaTeX document sometimes, and it's easier just to start writing. That's a good question. But yeah, I only use LaTeX for Drexel AI. Even my lab, my research lab at the Children's Hospital, we use Word. I know. Yeah. So I'll talk about lung mask here because I specifically did that, and then I'll talk about pre-processing genomic features. And I encourage people to do similar stuff, like just take a stake in one of the missing pieces of the paper, right? Because we want this to grow, and it was pretty shallow, but now it's getting deeper. And we had a problem with the citation, but I think that what was it?
I think Steve, or I forget his name, fixed it.
Where is his
Oh,
name?
Daniel fixed it.
Yeah. No, so that's good. So yeah.
Do you know who Daniel is, by the way?
He's a PhD student who works with Professor Kim in his lab. He's really good. Yeah, and he actually reached out to see how he could help, and I talked to him about different things. One of which is just doing a test to make sure that my reproducible code is actually reproducible. Cool. So yeah, code, paper, that's basically it. That's all there is to this project. We have a lot of background to do to survey, and I'm not too keen on doing the background. I can, but I'm not too keen on it. If anyone's interested in learning more about non-small cell lung cancer or radiogenomics as a field and what's been done, let me know. I did read 2 surveys that Savek posted a while ago. You don't feel like okay. Well, what you can do if you'd like to learn more, I think we posted 2 surveys. Alex, do you have them? Savek posted them a long time ago.
Surveys, I'm not sure.
Yeah, so Abby, specifically right here, there is this paper that he posted Thursday, October 21st. I'll get the title, which Dr. Yi, Alex, was Dr. Yi in the meeting last week?
No, it was Dr. I forget his name. It starts with a D. It's like it was one of the Penn researchers though. He was talking about us.
He was in the meeting?
Yeah, he showed up last time for like 5-10 minutes.
Cool.
And yeah, pretty much what we got out of it was that we might want to try looking at non-smokers and seeing how that would affect like things and correlations. Because he said there's not enough like research in that. So that could be a novel part of ours. Just a suggestion.
Sorry, he said, I don't, what do you mean
He's-
by that? He said,
He
look
said
at,
like look at non-smokers, the data for non-smokers, and see how recurrence predicts for them, or maybe how survival. Because he said there for like people who've smoked, and there's obviously a huge correlation between that and lung cancer, but people who don't smoke, I don't know. That's
you're
kind of what.
wondering what factors cause reoccurrence um, with non-smokers. That makes sense.
Yeah, because that could be a whole lot of different things like diet or exercise, or just anything. I think Satvik might be able to give more information on that, but yeah, he was pretty cool to talk to though.
Cool. It nice to get, another professor. Likely what happens with these papers that I send them over, or we typically send them over to Professor Kim to review them. He, leaves a few comments here and there, and then he gives us the okay to publish through archive. Through this paper. So yeah, Abby, I'm gonna post this one right here. And really what we need to do is have a survey. I think I talked last time. Do we still have the outline in the document? Yeah, we do. So yeah, we're going to structure it like this. We are setting up a motivation for looking at the disease, for using radiogenomics, and for using a specific model. And then the related work has to basically what has been done with non-small cell lung cancer. So that's like an umbrella, right, of all of the research with non-small cell lung cancer. What's like the best? What is the state of the art? Sorry, not state of the art. What are the endpoints they target? What are the features they use under that umbrella? It's a smaller umbrella. So for non-small lung cancer, what has been done with radiogenomics, and then has there been an evaluation of the model that we picked in literature? You could look at them like in that umbrella way, like I mentioned, or you could look at them separately and then just intertwine as you explore. Depends how you want to introduce things. I would first introduce the cancer, talk a bit about it, talk about radiogenomics, and then maybe talk about it in the scope of non-small lung cancer, and then talk about the model or the models that are used for radiogenomics or non-small cell lung cancer. You can kind of do that too. There's a lot of ways to structure that. There was a survey that I read. There were 2 surveys about radiogenomics, which were very helpful. I learned a lot from them. I wish I kept them. I'm just looking for them. Does anybody have any questions? Abby, did you want to ask something? I saw you turn your camera on. I turned it back off. It's fine if you did.
Oh, I just turned on my camera so that I don't know if my mic is working.
It is.
Oh, I just turned on my camera since you were talking to me.
Yeah, cool.
I didn't have to ask anything. Thank you, though.
There you got it.
Oh yeah, perennial. That's a good thing to bring up because Satvik, yeah, we were going to use transformers, which is still on the table, but Satvik also recommended there's pretty much an XG boost for convolutional neural networks as well as graph networks. Those are 2 other models we are considering using. So, I mean, I don't really have much experience with graph networks. So if anyone, do you, Ethan?
Graph networks.
Yeah.
No, I like graphs. I like how data is stored in graphs. I think graph theory is particularly interesting, good area of research, but I don't know graph networks. I actually just sat in on a presentation on graph networks. It was the first time I ever heard of them, and apparently I didn't understand it as well. But yeah, so here are the surveys, Abby, that I read like weeks ago, and it gave a really good overview of on radiogenomics, what kind of cancers. I think graph neural networks and transformers are two different things. I've used neither one of them, so it'll be really helpful for me to learn them.
Or if anyone just wants to like research the performance of them and like make a pros and cons list, that would help.
Yeah.
I think it's like a small task.
Yeah.
I've had good results with XGBoost, but I've not used the XGBoost CNN before.
I haven't either. Yeah. Like XGBoost was used for convolution neural networks, right? Did you mention another one for graph networks?
Yeah, so it's a graph neural network. So networks applied to, technically a neural network is a graph, but it's like a specific type of one.
I would try to explain this, but I don't know if I can. Actually, I can find it. I have a idea. We can make our own model, depending on how deep we want to do this and how novel we want it to be. But there's this paper that I read, or that we talked about in my last lab meeting on Monday at the Children's Hospital. And the idea is DNA methylation is a marker which is often regarded as being inversely proportional to gene expression. So if a promoter region of a gene is methylated, the gene is often turned off. Not always, but the idea is to predict the orange, which is the DNA methylation. So whether it's methylated 1 or whether it's unmethylated 0, using two things, the DNA sequence, so ATCG, and the surrounding methylation in like 500 base pairs or 500 nucleotides to the left and 500 nucleotides to the right. And what they did is they created this joint model to basically encode two types of features, the CPG or the DNA methylation features, and then the DNA features. The top one was using a bidirectional LCM because they're not dealing with a causal prediction. Basically, it's non-causal because this CPG, this position methylation is dependent on both what comes after and what also comes before. And then this one was just using a convolution neural network with like a VGG type architecture where it has the convolution and pulling layers. And then they connected into this joint model. So really what I was thinking is if we kind of take this idea and build a model that takes our features and then, specializes it. Like this one is specialized because it's for non-causal prediction. This one is specialized because it's using the convolutional filters that can be directly tied to like DNA motifs, which is like what proteins bind to. So we could try to do something like that because we have 3 different types of data that we're working with, right? We have the clinical, we have the RNA-seq, and we have the imaging data. I just wouldn't know the best thing to use for each one of them, but that's also an idea. If anyone, I won't put this in the GitHub, but if anyone wants to read into that, that's also there. That's just an idea. So cool. So this paper should be about 12 pages long, like in total, because usually when I write a paper, it's double columned, which comes out to be like 12, so 6 pages, and then single column pages is like twice that of double column pages about. And right, our title is taking up the entire first page, so we really should be able to get like 12 full pages out of this. I'll likely work on this over, I don't know, tomorrow night, and try to add in what I said I would. I would like people to volunteer for things, part of the paper, and this will be the highlight of the term. I'd like it to be I don't know, like a lot of work to be put into it. If you have questions about what specifically you could work on, just like message me, and I'm sure we could find something for you, and I can give you some papers to read for reference. Cool. Does anybody have any questions? No. I don't mind keeping this short. I don't want, I hate when the whole Zoom thing started, when COVID started, like everyone ambiguously said, oh, meetings are an hour. And I was thinking about that. I was like, why do they have to make meetings always 1 hour? It's never like an hour and a half. It's never 30 minutes. Everyone's just on for an hour and then off an hour. So we can make this one shorter. Cool. Thank you for your attention. If you have any questions, just message me on Slack. Cool. All right. I'll see you.
Thank you.
Peace.